Morning, morning.
Good morning. Professor Abbas, you can start now. Hello.
Hi, good morning, sir.
Okay, you can start.
Okay, sir. So good morning and welcome back to another day of online refresher course on Bioscience, Bioengineering and Nanotechnology. For today's first session, it is my pleasure to warmly welcome our resource person, Dr. Amit Kumar Mondol from Azad, Kolkata. So before we start our talk, let me share a brief introduction of our esteemed guest. So Dr. Amit Kumar Mondol is a professor in the Department of Biological Sciences at Azad, Kolkata. Dr. Mondol is an accomplished biophysical chemist whose work centers on structure-function relationship of disease-relevant proteins using cutting-edge tools such as advanced mass spectroscopy, molecular spectroscopy, and imaging techniques. His research bridges fundamental molecular discovery with clinical translations, focusing on major health challenges, including hemoglobinopathies, iron deficiency, anemia, cancer, diabetes, along with cardiovascular and kidney diseases. Dr. Mondol holds an MSc in physical chemistry from University of Kolkata and a PhD in biophysical chemistry from Bose Institute. He has previously served at St. John's Research Institute, Bangalore, and conducted research at IISc Bangalore and the University of Texas Health Center, USA. He is a recipient of several distinctions, including the prestigious Center of Innovation Award from Waters, United States. And it's worth to mention that this is the first Indian who get this award and only the second Asian scientist to receive this international honor. He has authored 66 publications with a strong citation records and with a patent and 3 book chapters. So, sir, we are delighted to have him with us today to speak on application of soft ionization mass spectroscopy in molecular medicine. Sir, we request you to kindly take the platform and enlighten us with your thought. So please, sir.
Thank you, professors, and very good morning to everyone. So let me share my screen. Select that. Is it visible? Hello?
No, sir.
No, sir.
Now? Okay. So
No.
It's not visible. Wait.
Dr. Shubhadeep.
Now is it coming?
Yes.
Yes, sir. It is coming now.
Okay. So it's clearly visible, I guess. Okay. So again, very good morning to everyone, and thanks for kind introduction. So let me start with my lecture topic today, application of soft ionization mass spectrometry in molecular medicine. I think the crowd is, hello?
Sorry to interrupt you, sir. It is your Gmail showing in front of us.
Gmail, okay. Well, let me close that. Oh, it's not the...
Sir, I think you selected a different window, sir.
Sir, you please reshare it. Stop this one and reshare your presentation window. Or maybe the whole screen.
Okay, wait.
Dr. Samanto.
No.
Yes, sir, I'm listening.
Now what's the situation? Is it...
No, sir. It is not coming, sir.
It's not coming.
Dr. Samanta, please ask me.
Sir, please go to the
share. Yes, that's what I'm doing.
and then you click on entire screen. Below that entire screen, it is
Enter
written.
screen. Yes.
Below that, it is written. Select a tab to share.
Yeah.
Then you
Then,
at the bottom.
yes. Now.
Okay, sir.
Now it's visible.
Sir,
Thank you.
visible.
Yeah. So let
Yes.
me go to slide presentation more. Is it now clear?
Now clear.
Yes, sir.
Yes.
It is clear.
Yes, sir. Yes.
Okay. Thank you. Sorry for the thing. So the topic is, as Professor said, that application of soft ionization mass spectrometry in molecular medicine. So there are two jargons basically. One is mass spectrometry and another is molecular medicine. With both the terms, some way or the other, some of us obviously familiar with. Mass spectrometry or mass of molecule, that we are learning this from our 5th, 6th standard. That is, any atom, any molecule or whatever exists in the universe, a particulate matter that has mass. So that's the mass we are going to talk about. It's a very fundamental property of molecular species. And molecular medicine obviously is a very broad subject. The very first thing comes in our mind is something related to the discovery of drug. No, that's not the case. Molecular medicine actually covers everything. It's a broad umbrella. It talks about anything related to human disease or disease in general rather, and that includes mechanism of the disease, molecular mechanism I mean to say, then obviously diagnostic biomarker and drug molecule so definitely is there. So we'll try to do one thing, that application of this mass spectrometry which actually measures molecular mass, and I'll try to help you out to just to make a link between these two. That means how mass spectrometry is helping to understand different features that comes under molecular medicine. So before I go to the main topic, mass spectrometry, let me start with very fundamental discoveries that is gifted to mankind in last century. One is urea. Discovery of urea made, that's the fertilizer, so it made possible for everyone on the globe to have food. Penicillin is another path-breaking discovery. I don't know how many trillion life it saved. And the other path-breaking discovery is X-ray, which made us possible to visualize inside our body without opening it. So molecular medicine actually started its journey not much ago. It's in 1949 by Linus Pauling, when he is the first person in his science paper actually made it possible to associate molecular event with human disease. And the one that he worked on is sickle cell anemia. So sickle cell anemia, I think many of us know, or almost all of us know, because government of India have taken an initiative in 2023, I guess. The government has announced that the nation has a health plan to eradicate this disease by 100 years of independence, that is 2047. Now the thing is, what he did, he actually tried to, in this paper, if you go through, you can make a note. It's a very nice paper, and that's the breakthrough, and that's the first paper in molecular medicine where molecular medicine started its journey. So many experiments are there. I'll just brief you only one or two. But before that, let me talk about sickle cell anemia. Sickle cell anemia was first discovered in 1907, I think, 07 or 08. It was first seen in US in the blood sample of a African student. And then it was just documented, but nobody knew what is the molecular basis. It was just seen that the sum of the red blood cells, which is biconcave in its nature, disc shaped biconcave, so that is taking some sickle cell. So that was the observation, and obviously clinician tried to associate that event with the clinical phenotype, treated and all. But nobody knew what is the molecular reason behind that. So in this paper, if I just copied one picture from this paper, which is down here. Can you see my cursor? Is it visible in the
Yes,
slide
sir.
itself?
Yes,
Yeah.
yes, sir. It
Okay.
is visible.
Okay. Okay.
Yes, sir.
So this is basically Longsworth scanning diagram, like gel electrophoresis, if I compare with. So the first one at the topmost corner, left topmost corner, is normal hemoglobin. So in this scan, normal hemoglobin looks like this. And the down one is from the patient sickle cell anemia. So they are movement-wise, if you look at the arrow, they are movement-wise, they are different. Then what he did is did the same scanning experiment, taken sample from patients with sickle cell trait. That means who are actually having some of the sickle phenotypes, or rather, if I be very specific, parents of sickle cell anemia patient. So their appearance is a bit different from the other two. And then what he did, a crazy experiment, that he mixed the blood in vitro, that is in test tube, normal and sickle, those two samples, and then ran the electrophoresis. And then appearance-wise, he looked, meaning he observed that the scanning is almost same as that of sickle cell trait. And from not only from this experiment, many other experiments, he drawn some very important path-breaking conclusions, which is what I listed here. So very first thing is he's saying that alteration in protein structure, and obviously protein structure, he's talking about hemoglobin, because hemoglobin is the main thing here, leads to disease. So he's first suspecting or noting that perhaps hemoglobin structure is causing, a structural difference is causing this disease, and this disease is inherited. That sickle cell anemia he is talking about is inherited. Third thing is, hence genes precisely determine the structure of protein, and one gene and one polypeptide. Remember, the most interesting feature is that in 1949, nobody knew the connection between protein and DNA. And at that stage, he's drawing this conclusion. Perhaps that is possible because the person is Linus Pauling, not a general one. Anyway, so scientific community had to wait 7-8 years to get to know what exactly wrong thing happened in hemoglobin. And it came in another path-breaking discovery, actually two consecutive paper came in Nature in 1956 and 1957 by Professor Ingram, and he could show that there is very minute difference in the hemoglobin compared to the normal one, and most likely one amino acid is mutated. So valine is mutated to glutamic acid. And if you who are having the biochemistry background, familiar with the structure, if you recall it, then the difference in the structure of valine and glutamic acid is very small. Only about a few atoms are different. So seeing the result, Linus Pauling's conclusion was that it's astounding that the difference in the structure is so small. Only about a dozen out of 10,000 are different. And on only such small atomies, man's fate depends. A person becoming disease, and another person is normal, and the difference is only few atoms. So that was the Linus Pauling's comment on the discovery of VM Ingram. So this is how molecular medicine started in 1949. Now I'll take you to the other jargon that I talked about is mass spectrometry. So mass spectrometry is there right from I think 1940s onwards, but biologists never been interested into mass spectrometry because the way to visualize molecules into mass spectrometer is that it's not that it can see neutral molecule. The first and foremost thing, bare minimum requirement is that molecule has to be ionic. And to make a molecule ionic from its neutral state, the way the treatment was that bombard with electron, high energy electron, so that it knocks off another electron, or fast atom bombardment. So all those ionization techniques were very harsh treatment to the molecule. And problem with biological molecule is that it's so large. Now, if you fragment it, if you make it into pieces, it's very difficult to reassemble those pieces and to find out finally what was your experimental molecule or what was the molecule that you started with. So that's why the biologists were not much interested in mass spectrometry until now, unless these two scientists discover a new method, that is soft ionization method, which is what there in my title, soft ionization mass spectrometry. So what it is, it's basically makes the molecule ionic without fragmenting it, very soft way. So one method that is called matrix-assisted laser desorption ionization, that was discovered by Koichi Tanaka in 1988. And another one is electrospray ionization mass spectrometry, that was discovered by Professor John Finn in 1989. And both of their discoveries were honored with the highest award, that is Nobel Prize in Chemistry in 2002. And if you look at the Nobel lecture, the title of the Nobel lecture by Professor John Finn is Electrospray Wing to Molecular Elephant. So molecular elephant, as I was telling you, the biological molecules are very large in size. So he's defining those molecules as elephant, and electrospray is a method that gives its wing. Why wing? Because to make molecule visualized under mass spectrometry platform, bare minimum requirement is that you have to take molecule from solution phase to gas phase, or solid phase to the gas phase. So literally molecule has to fly. So which is why he is giving the title electrospray wing to molecular elephant. Now, different type of mass spectrometry that I'll try to show to you in a short span of maybe 1 hour and 15 minutes is one. I'll tell you about proteomics. Proteomics is nothing but studying large number of proteins at one go. Okay, and that is mostly, as I'm going to talk about molecular medicine, so our interest would be on clinical samples. So literally to take the sample, whatever the biological fluid or solid sample, and then extract protein out of there, and then analyze them. Analyze using that ESI-MS platform, electrospray ionization platform, or multi-MS platform. And then this mass spectrometer to analyze this large number of proteins at one go is not easy job. So you need to have a front end part, which is a fractionation device. So generally, by and large, we couple liquid chromatography system with electrospray ionization. So that's why LC-SIMS. When it is nano LC, then it is NLC-SIMS. And we can use gel electrophoresis to separate out proteins. And then that is most effectively done by running the gel in 2 dimensions, so that you can effectively separate out large number of proteins. So 2D gel electrophoresis coupled to or combined to multi. In addition to that, I'll be talking about native mass spectrometry, isotope exchange mass spectrometry, and another one I'll talk about imaging mass spectrometry, imaging of molecules across tissue section, biological tissue section, focusing at proteins. Then we'll use multi imaging mass spectrometry, multi-IMS. And when we focus on small molecules like lipids, metabolites and all, then we will use DESI. This is desorption electrospray ionization mass spectrometry. Okay, so let me start with very brief introduction to the electrospray ionization. I said that soft ionization mass spectrometry, that means soft way to ionize large biological molecules. So how to do that? We just add little bit of acid when we want to make it positively charged, and we can take out a proton, acidic proton, when we want to make it negatively charged. And no matter whichever way the charge is, we actually do this in solution phase, and then we suck the solution through a small capillary. So whatever you are seeing as a large hose pipe, it's a very tiny small capillary. And then at the tip of which there is high voltage, 3 to 4 kilovolt. So when charged droplets containing, meaning solvent, mostly having volatile solvent or aqueous volatile mixture like water acetonitrile, and then you have the analyte, to that you added a little bit of acid to make it positively charged. So you are not doing any harsh treatment, you are not hydrolyzing any bond in the molecule, you are just having little bit of acid added to that, so that proton is sitting on the lone pair wherever it is available, on nitrogen, on oxygen and all. And then that droplet containing those molecular ions is pumped through a small capillary, at the tip of which there is high voltage, and moment it comes out, it sprinkles in air into tiny droplets. And then if you blow nitrogen gas orthogonally at 90 degree, then you are literally helping those solvent molecules to evaporate out. So eventually ions are coming closer, and at some point there is, when there is repulsion predominates between the ions, there is Coulombic explosion. So that eventually releases molecular ions into the gas phase, which is taken to the mass spectrometer, and then it's analyzed. Each and every molecular ion is analyzed into analyzer. So, and you get to see the mass spectra like this. I'll come to that afterwards, because that's what we are going to see all through the slides. So basically it has two axes. In x-axis you report m over z. M is the mass and z is the charge. How many protons are sitting there to make, when you are talking about positively charged by adding protons, so that each and every proton will add one charge. So that z comes here, and then y-axis is the intensity of ions. Now this is how the electrospray ionization work, and this discovery was honored by Nobel Prize to Professor John Fain. Now let me go to the other one, Koichi Tanaka, who discovered MALDI. So there again the concept is same. That means analyte mixed with organic acids, like these are the organic different acids, alpha cyanohydroxyceramic acid or dihydroxybenzoic acid, like that. And then you embed, actually that mixture you put it on a solid surface, solid metal surface, and then evaporate out, so that you have a matrix, and that is the organic acid, weak organic acid, along with analyte that crystallizes on the solid surface. Once that is done, then you fire the spot with laser, and then absorbing laser energy, matrix ionizes, evaporates out. Even analytes also evaporates out, and matrix, when it ionizes, it donates the charge, that is proton, to the analyte to make it positively charged, so that your job is done. Your job is done, and then you made analyte ionic, so that you can visualize under mass spectrometry condition, and then you record the spectra. So this is the spectra, and different ions that you produce, it passes through the analyzer. So to analyze them, each and every individual ions separately, ultimately it reaches detector, and detector generates this mass spectra. So this is a typical multi-mass spectra. So is it okay to understand this very briefly, if I give a pause of 10 seconds? Or there is any question? If not, am I audible? I don't know. Hello?
Yes, sir, you are audible.
Okay.
Yes, sir. Audible.
Okay. So
Yes,
let me go to talk about one important disease that we are discussing in this platform: hemoglobinopathy and thalassemia. So thalassemia, perhaps all of us know that they can't synthesize hemoglobin. So that's why thalassemia patients has to go through transfusion every alternate, say, once in a month or so. But hemoglobinopathy is, and in case of thalassemia, they can't synthesize hemoglobin. There are two globin chains in our hemoglobin. One is alpha and one is beta. So hemoglobin is made up of two globin chains in duplicates. It's alpha 2, beta 2. And thalassemia patients can't synthesize either of these chains. Like in India, especially in our eastern zone, we get to see thalassemia patients with who are devoid of beta. They can't synthesize beta globin chain. So that's how beta thalassemia is called. Now, hemoglobinopathy patients, what are they? They do synthesize hemoglobin, but they end up doing synthesis of wrong hemoglobin. And so that's why called structurally abnormal hemoglobin. And what is that structural abnormality lies with? There is a point mutation. One amino acid perhaps somewhere is changed. Alpha chain consists of 141 amino acid, beta chain consists of 146 amino acid. So in either of the globin chain, there is one mutation. That one amino acid may be substituted with another one. The one that I was given an example, sickle cell anemia. So sickle is basically a hemoglobinopathy, sickle cell disorder. So their beta globin chain at 6th position is mutated from valine to glutamic acid, one amino acid change, which is what discovered by V. Ingram I showed you. There may be deletion, insertion, or maybe fusion between two polypeptide chains. And if the worry is, if these things happen, then what's the fate? Yes, actually it's worrisome. And then the thing is, fortunately, I use the term fortunately, because if you look at the number of hemoglobinopathies discovered till date, it is, I think, crossed 1400 different types. So out of those, like in this crowd, all of us who are carrying normal hemoglobin in our body, we should feel fortunate. And then among these 1400 variants across the globe, not all are clinically violent, if I use the term. Most of them are clinically silent. So it doesn't bother day-to-day life. But there are few, like one example, I started with sickle cell disease. So that causes either unstable hemoglobin, altered solubility, so it precipitates, which is very common in sickle hemoglobin. And that hemoglobin precipitates inside RBC, which actually makes the RBC sickle in shape. Then there may be decreased oxygen affinity. That means it can't bind oxygen properly, or it binds oxygen more tightly. So in both way problem, meaning if it can't bind oxygen, then it cannot deliver oxygen in the tissue. So tissue will suffer from oxygen. If it binds tightly, then also problem, because then it doesn't want to release oxygen in the tissue. So there is again problem. And if you look at the statistics, though it's a bit old statistics, but the figure hasn't changed much. It's about 7% of total world population is affected by hemoglobinopathies. So 7 out of 100 patient, meaning 100 people, which is why I said that we are fortunate who are actually carrying normal human hemoglobin. So that means it's important to diagnose hemoglobin variant correctly. If I show you the alteration in oxygen binding affinity, let me show you the slide. Don't get bothered with too much write-up, because I use this slide in classroom teaching. You just focus on the right side, the picture here. So that figure, the red line is actually for hemoglobin, and the blue one is for myoglobin. So what it is x-axis is plotting fractional saturation and y-axis is PO2. If we remember just 3 years back, I guess 3 to 4 years back, when everybody were bothered about COVID, and then the parameter that we became very much familiar with, saturation, whether saturation has dropped or saturation is stable, something like that. And I don't know how many trillion oximeter has been sold in the market. So that's the saturation. So percentage of hemoglobin out of total, how much is saturated. So this curve literally tells how hemoglobin gets saturated with partial pressure of dissolved oxygen. And for biological assay, the parameter that is important is saturation of hemoglobin, 50% saturation of hemoglobin at a specific pressure, PO2. And for normal hemoglobin, it is 27 millimeter of mercury. That means 27 millimeter of mercury. So that much partial pressure of oxygen, dissolved oxygen, is required to saturate 50% of hemoglobin. Now, if you, why I'm referring this term? Because this is the PO2 value, P50 we say, P50, how much partial pressure of oxygen required to half saturate hemoglobin, that is used to define a hemoglobin variant, whether it is binding oxygen tightly compared to normal or loosely compared to normal. So I'll show you example like here. Again, you look at the figure here. So this line with red dots, that is for normal hemoglobin. Now you see the other one here in the middle, that is HBD Punjab, it says. So this is another variant. I'll come to what type of mutation it's having. This one, the other one which is having blue dots, that is HBE. This is another variant. Actually, in eastern zone and northeast India, this HB is highly populated. Now, all the whole point of showing to you is that if you look at P50 value, the P50 for normal hemoglobin is 27, and it got shifted to right. That means it can't bind oxygen compared to, or it's having decreased oxygen affinity, which is why you need to increase more of partial pressure of oxygen to make it half saturated. And for HBE is even more, it's somewhere around 40. So that means if there is mutation in the hemoglobin variant, that is what the patient of hemoglobinopathies is carrying, so their hemoglobin can't bind to oxygen appropriately. I'll show you another example like here. Here, this is for sickle. So for sickle also, it got right shifted. So sickle hemoglobin can't bind to oxygen properly. Here is another example which is left shifted, that cyan, but this is not hemoglobin variant, this is fetal hemoglobin. Remember, when all of us, when we were inside our mother, before our birth, so we used to take oxygen from our mother, and then obviously under that condition, under fetus condition, fetus stage, the hemoglobin should have more tight binding to oxygen, because the supply is very limited. We used to take oxygen from our mother, and then that's why it is left shifted. So that's why in very meaning even in 19 millimeter of mercury, it used to become half saturated. Anyway, so perhaps I have convinced you that how hemoglobinopathy functional assay differs from normal one, and why it is important. It is important from n number of disease, n number of cases. Like say if a child likes to take sports as activity, or when you are considering recruitment in army or in fire brigade or any armed force, so which needs very heavy physical duty as a regular task, so their oxygen should bind, hemoglobin should bind properly to oxygen. Say if a person can't bind oxygen properly, and if you send that person at the at the Kargil to fight for the country, then that would be disaster, because that person won't be able to breathe properly. So in many countries, this is mandatory. Before someone gets into army, hemoglobinopathy has to be checked, whether that person is a patient of hemoglobinopathy or carrying normal hemoglobin. But in India, it hasn't happened so far. So that's a separate story, though. Now, I'll show you how this is diagnosed in a hospital setting. In good old days, it was by alkaline gel electrophoresis. Same method used to use for thalassemia characterization. So basically, different hemoglobin bands appear in the gel, and obviously it should be read by an expert. And then the reading is if the band appears differently moving, like in this case, if you follow my cursor, this is normal hemoglobin. Other bands like here, this is fetal hemoglobin. This is sickle, D, Q, all appear in the same place. But the point is, it is different from normal. So that means if the band appears at a different place from the normal, so then that person is carrying hemoglobinopathy. Now, this method gradually replaced over more robust method is electrophoresis, liquid chromatography based system from gel electrophoresis. So here it is basically HPLC, automated HPLC. Biorad is one vendor, but there are many different vendors. I'm just citing this because this chromatogram is recorded in that machine. So this is how normal hemoglobin looks. HbA0, or sometimes we say HbA. This is HbA2 is delta variant, which we are carrying less, maybe 2-3%. And now, in case of variant, there is a peak, which is defined as HbA2. Like in this place, it appeared, but there is another possibility that HbE is there. Why it is? Because in general, HbA2 is less than 10%, but here the amount doesn't look like it is elevated HbA2, maybe different variant. So that's how it is HbE. But to characterize this as HbE, this chromatogram is not sufficient. We need to use other method. I'll show you another example where chromatogram came out with another peak, which is defined as unknown because the library of instrument doesn't have the information about this peak. So it just defines as unknown. So then, meaning clinical biochemist has to break his head to find out what it is, because patient is waiting outside paying appropriate fees to know the result. And then this is another one, which is a very interesting example. I'll show you in detail. So where HbA1c, that is the marker of diabetes, is showing as 52%, is exorbitantly high. It's not possible for a person to survive with glycated hemoglobin of that much. So this is the drawbacks of the existing method. Though automated HPLC is much better than gel electrophoresis here, but there are many such cases where variant sample doesn't have the appropriate answer. So the question is whether mass spectrometry can be useful, and that's where I'm going to link the mass spectrometry with one particular question that belongs to molecular medicine. Okay, so how hemoglobin looks? Hemoglobin looks here. This is basically LC. It's TIC, total ion chromatogram of hemoglobin. As I said that in mass spec we are talking about only ions, and front end part is liquid chromatography system, which basically separate molecular ions. So hemoglobin alpha chain, beta chain separates out, and this is literally mass spectra. Mass spectra where different I label different charge states. So alpha chain carrying 15 proton, beta chain carrying 17 proton, alpha chain carrying 20 proton, like that. So now the question is, what is the mass? Because it is protonated M by Z signal you are seeing here. Why x-axis is M by Z. But if you really want to know how much is the mass of alpha and beta globin chain separately, you have to deconvolute. So this is how deconvoluted spectra looks, and this is the deconvoluted mass. So alpha chain having 15125 Dalton, beta is having 15866 Dalton, and I think Dalton is the mass, meaning unit of the mass. Hydrogen is 1 Dalton, oxygen is 16, nitrogen is 14, carbon is 12, like that. So alpha globin chain has the mass of 15,125 Dalton, and beta chain is having mass of 15866 Dalton. By the way, this is the crystal structure of human hemoglobin: 2 alpha chain and 2 beta chain. There are 4 pyridine nucleus where oxygen binds. Okay, so now the question is how to use this platform to characterize hemoglobin variants that we are talking about. So this is basically proteomics platform. I'll not talk very detail about it because time will not permit me. So it's basically bottom-up proteomics approach. So you start with the molecule polypeptide chain, you digest, make it into peptides, and then those peptides we characterize and then build the entire sequence and ultimately build the entire structure of the molecule. So that's why we approach from bottom to up, which is why it's called bottom up. And here, when we are saying that we digest and make it small pieces, obviously then we measure those. So a long polypeptide chain will give you small peptides, now n number of, meaning large number of small peptides, and their mass we measure, ms. And we are not happy with just with measuring mass because there may be match with other protein also. So what we do, we fragment them. fragment into number of pieces. And that is the second dimension of mass, which is why it's called MSMS, twice MS. One is mass measurement of precursor ion, fragment it, and then generate daughter ions, and those daughter ion mass again measure, and that daughter ion is very characteristic of MS, meaning precursor ion. So there may be match of mass of precursor ion from two different proteins, but daughter ion structure would be different. So it's very important to have the MSMS spectra recorded to ultimately build up the sequence information. So I'll quickly, this is MSMS. Okay, I'll quickly skip through this. Okay, so this is though complicated, but just focus on whatever I'm requesting you to look at. So this is what the HPLC trace of sickle hemoglobin looks like. This is variant. Now, in mass spectrometry platform, if we want to characterize, we have to digest into small pieces, generate a large number of peptides. So those peptides actually get separated in the front end part, which is nano LC-SIMS. I was mentioning you NLC-SIMS. So this is NLC part. And then each of these peaks consist of one or multiple peptides that you characterize both in MS and MSMS mode, and ultimately get to know the sequence. And everything happens in background, this database search, and ultimately you are interested to know which variant it is. So like here, it pops up with the variant. Here it is hemoglobin subunit beta, HBS, and then this mutation is glutamic acid, a single letter code, glutamic acid to valine, right? That is the mutation. In normal human hemoglobin, we are carrying glutamic acid, and that got mutated to valine, which is what it happens in the sickle. Okay, and this is the MSMS spectra. So finally, we get to know the point mutation. Now I'll show you some example where our conventional method didn't give the right picture, like HBD Iran. So this LC profile that is used in hospital diagnostics, it came up with some higher percentage of HbA2. As I was telling, that normal person doesn't carry this much HbA2. Anything more than 10% is suspicious. So here it is 21.3%, and through mass spec, you get to know that it is actually a not normal hemoglobin, not HbA2 delta hemoglobin. It is actually a mutant, beta mutant, HBD Iran. Mutation has happened at the 22nd position, glutamic acid to glutamine. One more example, again huge elevation of HbA2, 63.5%. And if you characterize this, it is HbE. HbE, that is at the 26th position of beta globin chain, glutamic acid to lysine. Another example, HBD Punjab, I was mentioning that. It didn't give any specific information. LC trace in hospital diagnostics gave you unknown. But if you take this sample through mass spectrometer and characterize them, then you'll get to know the answer. It is HBD Punjab, mutation at beta position, glutamic acid to glutamine at 121 position. Another example, the last one which I was mentioning to you, that it is very interesting, HbA1c, that is the marker of diabetes. In addition to glucose, I'll come to that. Another good marker is HbA1c, glycated hemoglobin or glycosylated hemoglobin, that doctors prescribe to measure. And then recommended value for, if a person is normal, that HbA1c is less than 6%. Pre-diabetic person has the range 6 to 6.5. Diabetic person has value more than 6.5. And this fellow is carrying, at least LC is showing that 52.1%. And this lady actually came to the hospital just for buying insurance. They have to go through different medical tests, that's all. If you look at the blood glucose level, pretty normal, but HbA1c value traced from the conventional LC profile is exorbitantly high. And if you actually, we characterize this variant using mass spectrometry platform, and she is absolutely fine. She is basically carrying a variant, which is benign variant, HB Beckman, mutation at beta globin chain at 135th position from alanine to aspartic acid, A to D. So this is how the mass spectrometry platform could be useful in molecular medicine, at least for diagnostic purpose. That's what I tried to represent in front of you. Now, here is the thing that I was telling. For a person, like this zigzag line is showing how blood glucose level in a person's body varies across the whole day, and it is recorded over 4 days. It depends on food intake and exercise also. And if you look at this zigzag line, that it indicates that the blood glucose varies a lot over time within a day itself. But if you look at this middle line, which is pretty straight, it doesn't vary. And this is basically your HbA1c value, HbA1c of 7, which is why HbA1c is a good marker, because it doesn't change. Generally, the psychology of a patient is that before going to go to hospital to get the blood glucose level check, and then meaning getting diagnosed whether I am diabetic or not, is reduce the rasgulla content and then do exercise a lot and then go and make the clinician full. That you perhaps make it possible by glucose, but not by HbA1c level. So, which is why HbA1c is very important to characterize. And I'll show you how mass spectrometry can be helpful in that. Yes, so this is the chromatogram where in this is again LC stress, where HbA1c, this red peak, actually gives the value. That's how in hospital, everywhere across the world, hospital, it is calculated. Normal is less than 6, pre-diabetic 6 to 6.5, diabetic more than 6.5. Anyway, so multiple questions can arise in your mind, as you have seen now that hemoglobin variants, because in addition to normal hemoglobin, there are 1400 possible variants. So what about glycation of them? There is no such point that if a person is hemoglobinopathy patient, he or she will not be become diabetic at some point in the life. No. So if their glucose adduct, basically HbA1c is a glucose adduct. So that glucose adduct will happen, and then question is, where do they elute? In the LC trace, if they don't elute in the place where HbA1c eluting for normal, then they'll be underestimated. Okay, so that is one problem. So question may arise, HbA1c, whether it's a good marker for hemoglobinopathy patient, even for poorly controlled diabetic patients, like one that I am raising the question here, who is having very uncontrolled diabetes, doesn't listen to the clinician, doesn't care about sweet intake, don't exercise, and all those sort of patients. Their glucose adduct, see, it's basically chemical reaction. Glucose is reacting with hemoglobin molecule. At primary amine group, it's getting glycated. So primary amine group is there plenty in number in hemoglobin molecule and across any protein as well. So it's not only one specific glucose, one specific primary amine will be always glycated. It's expected that you may have multiple glycation also. And if multiple glycation, where do they glycate? And ultimately where they elute out in the profile, and if they elute out not under this peak, then they will be underestimated. So the question may arise, is it still a good marker in case of poorly controlled diabetes? We'll see to that. And as I was telling, that whether in case of hemoglobin variant, the question may arise whether glycated fraction of them will elute along with HbA1c for normal. Don't know. And then another question is, as N-terminal alpha amino group is glycated in the HbA1c, beta globin chain N-terminus, N-terminal NH2 group, that gets glycated. That's how this HbA1c molecule is. But there are many, but because of this point mutation, structure may be different. Accessibility of those NH2 group may be different. If that happens, then who guarantees that any variant will have equal susceptibility or equal accessibility to glucose to get glycated? So that is another question. So lot many questions are there to answer, and very till today very poorly, meaning very little is known about those. Anyway, so I'll come to answer some of those questions using mass spectrometry. Another stage or the disease status, that is oxidative stress. We all of us know about this term nowadays, oxidative stress. It's linked to n number of disease, starting from blood glucose, diabetes to hypertension, whatnot. So basically, nature has given a molecule glutathione, which is tripeptide, which actually an antioxidant, natural antioxidant, all of us carrying in our body, and that fights with the oxidizing agents, xenobiotics. So that may be free radical, that may be hydrogen peroxide, superoxide radical, oxide radical, and all. So this glutathione's job is to kill those oxidants by the way that GSH, glutathione, gets into GSSG. All of us know from our childhood chemistry that if in oxidation reduction reaction, if someone gets oxidized, then some other fellow has to be reduced. So when glutathione is actually reducing oxidant, so it has to get oxidized by itself. That's what it happens, GSSG. Now, one of the fate of GSSG is that it undergoes thiol exchange with protein, like thiol exchange with hemoglobin, forming glutathione hemoglobin. And if you think of a continuous stress, then the glutathione hemoglobin content in the body will be elevated. And that's how the question is, then, is that molecule could be a marker of oxidative stress? Let us see that. I'll cite one one case, renal failure case. So before I take you to that result, I'll just quickly show you how alpha and beta globin chain appears for normal subject. I showed this type of spectra to you at the beginning. That was a bit colorful. This is black and white, but it's safe spectra. And then it's you focus on deconvoluted stuff. Alpha globin chain, glycated alpha, beta globin chain, glycated beta, glutathione beta. Now, we have taken three category of patients. Meaning all are renal failure, their kidney doesn't function anymore. One category is transplant patient, where kidney transplantation happened, and after that they are stable. Two other category are dialysis patient. One is hemodialysis patient and another is peritoneal dialysis patient. And if you look at their alpha globin chain status, so this is glycated hemoglobin and glycated alpha globin chain, this is normal alpha. So these are the three patient group. And then beta glycated, beta glutathione. So if you look at, I'll take you the previous slide, there is no glutathione alpha, but there is glutathione beta. So glutathionylation happens only in the beta globin chain at the cysteine 93 position. Now, I'll just quickly skip this. Yes, now the question is, can we quantify it? Yes, we can quantify through mass spectrometry. We literally measure the peak height and then try to correlate the intensity with the concentration. And then that's how the glutathione hemoglobin or GSHB is calculated. And if you don't look at all this crowded slide, if you look at this figure, this line first, for normal group of patient, some value is 3.15. That is the percentage of glutathione hemoglobin present in a normal sample or normal person, healthy normal individual. Transplant group, kidney transplant group, peritoneal dialysis, hemodialysis group. So if you look at in this category, it's gradually increasing, and that has to be there, because in case of both dialysis patients, the toxic nitrogenous substance which we can't excrete, meaning we are excreting who are having no kidney disorder so far, but these patients, their kidney doesn't function, so those nitrogenous stuff is getting accumulated in the blood sample. And then when dialysis is happening, the this entire blood is coming out and then going through dialyzer, so it's exposed to the foreign surface. So that is this uh, place of stress, and then that stress is expected to be highest in hemodialysis group, which is why you see here. So literally the message that I want to give it to you is that we have not only for chronic renal failure, we actually measured for other category as well: iron deficiency anemia, major depressive disorder, psychiatry patient, and then multiple sclerosis patient. So everywhere glutathione hemoglobin is up, and all these patient categories are very pretty well known to be under oxidative stress. Other group of people across the globe have shown that diabetes, Friedreich ataxia, cardiovascular disease, hyperlipidemia, in all cases, this molecule is elevated. So that means glutathione hemoglobin is elevated under oxidative stress. So it could be a clinical marker of oxidative stress measurement, which hasn't come to the diagnostic so far, but yeah, maybe in future possible. But one message I want to give it to you: it has been observed that sickle hemoglobin, which we were talking about, if that gets glutathionated, then sickle cell polymerization gets inhibited. So the question is it possible for sickle cell patient that glutathionylation is a therapy? No, because sickle patient is already under oxidative stress, and to elevate glutathione hemoglobin in any person's body, you need to raise oxidative stress. So you cannot make is meaning to cure one thing, you can't make you can introduce another disease. So that's not acceptable. Anyway, so I'll take you to other mass spectrometry, native mass spectrometry. So native mass spectrometry is a different type of one. So far I was talking about where we were looking at the different polypeptide chain, where our three-dimensional architecture is lost. We are seeing alpha globin chain separately, beta globin chain separately, and then when we are talking about hemoglobin spectra, we are not seeing tetrameric hemoglobin, alpha to beta 2, rather alpha beta separately. Now question is, can we look the entire intact molecule in mass spectrometry platform? Yes, it is possible, and that is done through native mass spectrometry. So in native mass spectrometry, you have to gently ionize the molecule and take it to the gas phase. So here is a spectra, native mass spectra of normal human hemoglobin. So here 3 different charge states. So these circles, 2 red circles and 2 blue circles. So one is for alpha, one is for beta. As I said that it's alpha beta in duplicate, so 2 alpha, 2 beta are there. So these are the different charge states of normal human hemoglobin, and this is how its mass appears. So you can get to see the mass of intact protein molecule where multiple globin chains or multiple polypeptide chains are bonded together by non-covalent interaction. Okay, so how to make sure that whatever I am seeing is from that molecule? You very simply fragment and you generate the individual chain. Like you take the this tetrameric molecule and fragment it, you will get to see the alpha beta, which we are now familiar with to see. Okay, so Beckman, the one that I was explaining to you, that lady appeared with exorbitantly high HbA1c content. Actually, she is not carrying any that sort of glycated hemoglobin. Her mutant hemoglobin that elutes at the same place where HbA1c elutes. So that is what is very important information, because if a person carries Beckman variant, so it can, meaning it can fool clinician saying that they're exorbitantly high glycated hemoglobin. No, that's not the case. And if you look at the blood sample through native mass spectrometry, that's how it looks. It's beta variant and beta normal, blue and green one. So the tetramer is having 3 different population. One is normal hemoglobin, 2 alpha, 2 beta. One is variant hemoglobin, 2 alpha and 2 variant beta. Both the beta chains are variant, and then mixed one. That means 1 alpha beta is normal and 1 beta is variant. Anyway, so now coming to the glycated case. So this is a glycated fraction of a poorly controlled diabetic patient. HbA1c is 19%. Very difficult to survive in this zone. So if you look that person's glycated fraction through native mass spectrometry, you'll be surprised to see that it's basically carrying all sorts of glycation. Normal, this is normal, trace amount of normal, because we have separated the glycated fraction from normal. Here it is tetramer with 1 globin chain glycated, 2 globin chain glycated, 3 globin chain glycated. So all possible glycations are there. And if you look through mass spectrometry in bit rigorous way, by the way, these are different dimers. So monoglycated hemoglobin, diglycated hemoglobin, triglycated hemoglobin, if you fragment, so monoglycated, when I'm talking, that means hemoglobin carrying 1 glucose. Diglycated meaning hemoglobin carrying 2 glucose. Triglycated hemoglobin carrying 3 glucose. So if you fragment them, their pattern is same. That means when this molecule is getting glucose into it, then it can go either to alpha or to beta. In within our body, it's happening equally, which is why you get to see alpha chain is glycated, also beta chain is also glycated. Meaning you started with tetrameric hemoglobin with 1 glucose, but when you are fragmenting, then you are seeing that both the globin chains are getting glycated along with their normal fraction, definitely. So how it is possible? It's possible if some of the tetramer having glucose sitting on alpha and some of the tetramer glucose sitting on beta. That's how you can generate. And the scenario didn't change in terms of daughter ion spectra when you see at the diglycated species. Same spectra. If you look at the different labeling, it's same. And that is possible when the glucose, meaning second glycation, happening on the globin chain, which is not glycated. So nicely is maintained within our body. You can't do this in a test tube, not possible. So it means that when second glucose is coming to glycate the hemoglobin molecule, it's preferring to go to that globin chain which is not glycated previously. And same thing is happening, repeating when for the third one. Okay, so that means glycation of hemoglobin happening symmetrically across alpha and beta globin chains in vivo. If you literally interested to measure the glucose content or glycated individual globin chains, as we done, I showed you in case of glutathione. So you can literally calculate the glycated alpha, glycated beta, and as tetramer is consisting of 2 globin chains, so you just sum them up, divide by 2. That's how you get to know percentage of glycated hemoglobin as such. I'm talking about tetrameric hemoglobin. And then this is how the different values of glycated hemoglobin measured through mass spectra. And this is in the left side, left column, you have glycated hemoglobin measured in conventional HPLC. That's the machine used in diagnostics in hospital setting. Fortunately, they are actually correlating nicely. And that's happening because the glycation within our body is happening symmetrically. So the question that we had in our mind, when there is multiple glycation in globin chain, in glucose, in hemoglobin molecule, multiple glucose are sitting there. Is it still HbA1c measured through hospital diagnostic tool is trustworthy? Yes, it is trustworthy because the glycation happens symmetrically. Okay, now so this is something that I tried to correlate with. So the thing is, diabetic population is exponentially increasing in the across the whole world. So it is defined as a non-infectious epidemic in the present century. And if you see the BMI, generally it's better for us to try to be within normal weight by monitoring our food intake and doing exercise also. And I'll end this slide with a nice quote that I saw in a corporate hospital. It's actually predicted that out of 4 people, 1 would be diabetic by 2030. So the quotation is, if there are if 3 of your friends are not diabetic, better check yours. Okay. So let me come to the sickle cell anemia story. So sickle cell anemia, as I was telling, that it was detected in West Indian student in Chicago first, but molecular mechanism was not known until unless Linus Pauling discovered that it's meaning it's a molecular disease, and from V.N. Ingram's discovery, it was glutamic acid to valine at the 6th position of beta globin chain. So by now it's pretty well discovered how the this this polymerization happens. See, this picture shows sickle cell RBCs, and this is normal RBCs. So you see the entire shape has changed. Why it does happen? Because tetrameric hemoglobin associate. So this picture, if you look at, the encircled portion is basically showing one portion of a molecule is inserted into another in a groove of another molecule. So the hinge portion is contributed by the valine residue, isopropyl group of valine residue. So I said that glutamic acid is mutated to valine. So in normal hemoglobin, this residue is not there. It's meaning it's not valine, it's glutamic acid. But moment it is valine, then that valine has this isopropyl group which is protruding out from the surface, and that gets into the groove of another molecule, and then it's called basically lock and key mechanism, so it becomes associated. So this is the initiation of polymerization. How it goes on, so it's actually hydrophobic patch of one molecule binds to the hydrophobic group of another molecule, and the hydrophobic group of this molecule binds to the hydrophobic patch of another molecule. Hydrophobic patch of this molecule binds to the hydrophobic group of another molecule. So that's how the polymerization proceeds. So ultimately, this chain forms. Okay. So consecutive addition of HBS tetramer aggregates results double-stranded polymeric chain. Once this double-stranded polymeric chain happens, then it actually association of 7 such double-stranded polymer forms fiber, and then that fiber ultimately is obviously has to be stabilized by other interaction, which is intrastrand as well as interstrand, and then ultimately it precipitates inside cell, and that's how it makes the cell as sickle cell. So that's the disease is. And all these phenomena, whichever I showed you, it happens when the molecule is under deoxy state, meaning oxygen has been dumped into the tissues, hemoglobin is free, and that's the deoxy state. Until unless it comes back to the lung, it cannot be oxygenated again. And this phenomena happens, this phenomena starts there itself. Okay. So what's the fate actually? It gets clogged. The moment this sickle cell is there, so then it can block the capillaries, and that's how the patient gets into stroke, heart attack, knee pain, or all kinds of joint pain, breathing trouble because it cannot carry oxygen anymore. So oxygen deficiency is there. Patient is suffering from hypoxia, so everything starts. And there is a definitive time, delay time. This association happens, and until and unless this large number of associated molecules combine together, this polymerization doesn't proceed. So that time is defined as TD, delay time. There is a way to calculate. I'm not going to go to that, but all I'm going to say to you that this delay time is very much concentration dependent. Now, in case of sickle hemoglobin, 10 micromolar concentration in vitro I am talking about, in test tube. So the delay time is about 47 seconds. But as I was telling you, glutathione has been observed to be helping in the way that it prevents polymerization. So if this same concentration of glutathione hemoglobin, if we take, then the delay time increases to 188.26 seconds, almost 3 minutes. What's the advantage of that? See, I said that it happens, this phenomena happens only in deoxy state. So moment oxygen is dumped into the tissues, it becomes deoxy, then event starts it. Now, it takes time, again, blood takes time to come to the lung, and then oxygenated. Moment it is oxygenated, then this polymerization doesn't happen. So this event of delay time, if we increase this delay time by any means, then it's possible to prevent, because if we increase the delay time, by the time hemoglobin comes to the, by that time hemoglobin comes and reaches to the lung, it gets oxygenated, and there is no fear for getting polymerization again. So that is why delay time increase is very important. Many people across many pharmaceutical industries across the globe is trying to increase the delay time by using different molecules. That's how different drugs are discovered, but till not very successful has happened, meaning successful event has happened with any particular molecule, which is why the drug is still not there in that sense. It's only possible to manage to some extent, but obviously in future it will come. I'll cite one example that I got a sample from one patient which appeared in the AIMS Kalyani Hospital, with whom where I have collaborator. We are just outside the meaning other side of the road located. So there one patient is appeared with sickle cell, but and sickle percentage, if you see, is pretty high, 50%, but that person didn't have any symptom, meaning any problem with whatever convenient, meaning which are familiar, which are common for sickle cell anemia cases. It was very surprising. And if you see, then the fetal hemoglobin is a hemoglobin which we were carrying when we were in the fetus stage, that is inside our mother body. The moment we took the birth, that fetal hemoglobin, that HbF shuts off, and normal hemoglobin which we are carrying presently actually starts getting produced. So that patient is still expressing large amount of fetal hemoglobin, 40%. And very rare such case. I have, in my entire 25 years career with hemoglobinopathies, I have seen perhaps 2-3 such cases. And if you look the hemoglobin through native mass spectrometry, it's very surprising that the molecule is actually consisting of different type of combinations. Like in case of Beckman, I was explaining you, here also beta sickle combined with fetal globin chain and 2-alpha there. So this is mixed population, and this mixed population will not have this elongation of the polymer chain happens. You remember, if you meaning if I take you to this slide, this association is very much crucial. Okay, let it happen between two molecules, but then it has to go, and meaning chain wise it has to happen. Now if a tetramer incorporates fetal chain, like in this case here, then perhaps that elongation will be stopped, and then polymerization will not proceed. So this is why it's happening, meaning this is perhaps what's happening in this case. But not only in case of so the question may pop up, then what if we can increase fetal hemoglobin chain in patients of sickle cell anemia? Yes, then it's possible. Actually, that's how a whole world is running behind, not only for sickle cell patient, but in case of thalassemia patients, because whoever is not carrying, not producing beta globin chain successfully, if fetal hemoglobin starts, gamma hemoglobin starts getting produced, then it's possible to lead the normal life without any transfusion and all. But nobody has been successful in switching fetal hemoglobin synthesis in a adult body. So this is again $1 million question, but hope if that happens, then people will be benefited out of that research. I'll take you quickly through another type of mass spectrometry, hydrogen deuterium exchange-based mass spectrometry. So this is basically we exchange polar hydrogens with deuterium, and deuterium being isotope so of hydrogen. So there should not be any change in chemical property, but you add 1 Dalton mass, because hydrogen is 1 Dalton, deuterium is 2 Dalton. And moment the mass is changed, you can trace it through mass spectrometry, and then you come down where that change has happened. So then if you can look at that, then you can talk about the conformation of the molecule, conformational change that happens with the molecule. Out of time, I'll quickly go through it, because I think I have only 15-20 minutes left. So I'll not take you through these details. Rather, I'll just show you one example. Say sickle hemoglobin. This is the groove, right? And this is the hinge. Now, this association I said, this happens only in the deoxy state, not in oxy state. So question may pop up that why doesn't happen in the oxy state? Okay, so best thing to see the crystal structure of oxysickle hemoglobin. Unfortunately, that hasn't been discovered till date. I don't know why, because I'm not a crystallographer. I don't deal with the molecule to crystallize, but it hasn't happened. Now the question, the very simple idea is, like, if I talk about this groove, and if it is very essential to exist in deoxy state, maybe in oxy state it's not there, the conformation has changed. Now if you look at this groove region, here the solvent accessibility is less. So the exchange that I am talking about, hydrogen and deuterium, that would be less in these cases if the groove exists. And if groove doesn't exist, if it becomes flat, accessible to the solvent, then that exchange will not happen. That exchange will happen at a much faster rate. So that may be a reason, and that's what we observe, that yes, in case of oxyhemoglobin, something like that happens. This region, hydrogen deuterium exchange rate becomes very fast. I'm not taking you through the details of the result for the time constant, but that's what we observe. Not only that, even we have seen that, see, this group, this hinge contributed by valine, it has to be stereospecifically oriented in space so that that association happens. Now in case of oxy, this portion becomes highly dynamic, so it's very difficult to take this groove and then make it to sit into the take this hinge and make it to sit into the groove. And that is another reason. There are other interactions also which you have seen part out, and these results were published in this journal, which you actually got honored by cover page also. So you can make a note of this publication if you're interested to know about this event. Anyway, so the message is that using hydrogen deuterium exchange, not only the sequence but also functionally active structure, functionally active conformation of a molecule could be monitored. The change in conformation associated with a biological event can also be monitored. Yes, this is another event, so I'll not go into the detail, but all I'm saying is that we can insert D2O inside a live cell, which is what I did, and then make this exchange happen inside the cell. So you can literally look at the structure of a molecule inside a biological cell, meaning live cell, where it is extremely crowded with many different type of molecules. Okay, so using mass spectrometry platform, we can literally calculate the thermodynamic parameters also, dissociation constant or association constant. That is also possible. How I am not taking you through, but I am just giving an example that using native mass spectrometry, it's possible to calculate KD, which is what I have done here. So for normal hemoglobin, it's about 1.81, and KD2, then see it is tetramer. So tetramer to dimer, one dissociation, dimer to monomer, another dissociation. So KD2 refers dimer to monomer, KD1 tetramer to dimer. So upon glutathione, there is fold change in this dissociation. I'll talk about cigarette smoke related issues, which is again another health issues. So basically we have seen that this cigarette smoke is having 4,000 compounds in it, and quite alarming. One of them is parabenzoquinone, which is very highly reactive. Whoever from chemistry background, they know very well, better than me perhaps, that parabenzoquinone or any quinone, how strong their electrophilic nature is. They snatch the electron wherever it is available, and that's how moment it is into our body, it actually conjugates. This curve basically shows that how parabenzoquinone conjugate with hemoglobin increases with number of cigarette smoke per day. Okay, so how it happens? Parabenzoquinone, actually in tobacco it is parabenzosemiquinone, which gets oxidized to parabenzoquinone and it forms conjugate. And then if you look back, the oxygen dissociation curve, so that parabenzoquinone adduct that is left shifted, it binds to oxygen very tightly. And if you look at the dissociation constant, then there is significant change. Tetramer to dimer dissociation increased by 5-fold from 1.81 to 9, and there is almost 60-fold increase for the dimer to monomer dissociation. So literally hemoglobin is breaking down upon this conjugation, and moment it breaks down, then it cannot dump oxygen successfully to the tissues. It generates hypoxia, and as you know that upon cigarette smoking for decades together, people actually end up with one of the diseases is COPD, chronic obstructive pulmonary disease. Okay, so the thing is, wait a second. Now the thing is, so this could be a reason of hypoxia in addition to all other mechanisms which has been established over the years. I'll just show you how dangerous it is. See, this is this picture I have taken from net, Google picture, some publication somewhere. This is how normal RBC looks, and in case of smokers RBC, this is like the case. The one that I have done in our lab, this is scanning electron microscope picture. This is normal RBC. Now you see, moment parabenzoquinone is forming adduct. I'm not talking about hemoglobin adduct. It can adduct with anything in the world. So it's actually making modification at the surfaces also, and then this surface becoming bulging. There is bulging here and there, and that is problem, because moment nature has given this disc shape molecule, biconcave disc shape, so that it can squeeze through capillaries, fine capillaries. But moment this bulging happens, we have measured through fluorescence anisotropy. You can literally measure whether the flexibility has changed or not. So then flexibility is changed significantly, and that perhaps causes again kind of blockage similar to that we found in case of sickle, and then perhaps that might lead to stroke and whatnot. Okay, so last one, I'll quickly finish. I have come to the last part of the talk. This is imaging mass spectrometry. So imaging, tissue imaging is a hardcore event in a hospital setting, in a path department. So surgically removed tissues are taken to the path department. They are sectioned and then stained, H&E stain, hematoxylin and eosin staining is done. And then pathologist's duty is to check that stained tissue section under microscope, and then come to conclusion whether the patient is carrying any cancerous cells, tissue is cancerous or not. Now, remember, someone this is very crucial in case of cancer, meaning removing cancerous tissues, which is what the job of oncosurgeon is. So while removing that, oncosurgeon doesn't know whether I'm removing correctly absolutely everything. So that's why he sends the sample to path. Quick H&E staining is done because patient is open on the OT, and then information comes back to him from pathologist whether I have cut successfully or I need to cut more. So that surgical margin is very important in defining. And remember, one person is looking at the slides physically. So this false negative rate is, though it's very less, but it's still there, 2.4% according to this report. Though this is gold standard method, it's well accepted, followed across the globe, but there is not zero error. So moment some of the cells are left, that's how the relapse of the disease happens. One reason. Immunohistochemistry, staining with antibodies, but there is again limitation because non-specific binding. Okay, so the question is, can there be any method which will give zero false positive result, zero false negative result, so nobody will suffer from relapsing it, at least for this reason. So their mass spec may be an answer in future. So I'll quickly show you two type of imaging mass spectrometry that I have proposed in the beginning. One is using multi, where we actually section the tissue, then we process it and remove basically salts, lipids, then digest on situ itself, and then fire with laser. There also we fired with laser, but here the difference is while firing, laser knows which coordinate it's firing. So that means XY direction is known. And then this is an example. That means basically from this tissue section, different proteins has come out at different place. That is obvious because biology is very complicated. Tissue is heterogeneous. A protein molecule, every single protein molecule is heterogeneously distributed across the tissue section. So then different portions give tissues, meaning proteins, image differentially, and then the job is of mass spectrometrist is to combine to form back the image. So basically you visualize tissue section in terms of molecular population. And I'll show you one example, prostate cancer, that is commonly found in case of 80% men over 60 years. I'll not go through the details. I'll just show you. So this is different method of detection. PSA, that is the blood test, more than 4 nanogram per ml of blood, then it is prostate cancer. Less, it is less, but it has been found that there are cases where even PSA being less than 4 nanogram per ml, the prostate cancer has already developed. So that means we need to see a good marker. So I got this prostate cancer tissue image done in Tata Cancer Hospital in Calcutta. So this tissue basically filled with many cancer cells. So this is how cancer cell looks, and this is what the MD Path person's expertise is to read about. I have learned little, but because of time constant, I'll just skip through. I'll come to image. So this is basically same tissue section, not same, serial section of the tissue, same tissue. And then its image. This is how one protein distribution looks across the tissue section. And if you look at the molecule, it is basically myosin, which is a cytoskeletal protein. So it is expected to be everywhere there, irrespective of cancer or non-cancer, meaning normal. So that is what we found. Now I'll show you another example where this is one particular population, meaning area, where one particular protein measured through image or detected through image has been found to be heavily expressed. And if you characterize that protein, this is vinculin. And then vinculin, what is its function? Its critical component adhesion between a cell and the extracellular matrix. Remember, in cancer cells, the moments it migrates from one organ to the other, that cell has to adhere to the other organ, other tissue, and that adherence, this particular protein does the job. So if you compare the in the histopath image, in this region it's filled with cancer cells. So there is nice correlation. I'll skip through this. I'll just end with another important last topic, that is DESI, desorption electrospray ionization, where instead of protein molecule, we actually image small molecules. Like here it is again prostate cancer tissue. I have taken this picture from this paper, PNS paper, which is somebody else. And then as per histopath, this portion is cancerous and this portion is normal. Now, in case of the difference between previous one and here is, here you, instead of laser fire, you fire with a jet of solvent with varying polarity, so that you take the small molecule out of the surface and then you image them. So you see here different molecules, lactate, succinate, the erythrose, glutamine, glucose, citrate. So they are how they are distributed. Some of them are distributed uniformly, some of them are distributed in the cancerous zone predominantly, and some of them are in normal zone. So they have actually, in this paper, their nice finding is they have focused on two metabolite, glucose and citrate, and they have seen that the glucose over citrate ratio that changes dramatically when you move from normal zone to cancer zone. I'll tell you where. So this is normal and this is cancer. Now this rate indicates that the ratio is more than 1. So how you see that in the cancerous portion it's heavily, meaning this ratio is high, and it's same all over all the other prostate tissue wherever the histopath defines as the cancer. And that's what the conclusion that they are drawing, that across the surgical margin which I was talking about, or across the margin between the normal and cancerous cell, how the ratio is changing. Citrate is dropping and glucose is going up. And if the way they are referring is, wherever this glucose citrate ratio exceeds 1, that is the cancerous zone, and it is, if for normal it is less than 0.5. So this particular device is going to come, as per my prediction, to the diagnostics very quickly. It has, prototype has been developed by somebody. Actually, Desi was discovered by Graham Cook in 2004. One of his postdoc, one lady, she actually invented this pen, mass spec pen. So here, basically, you see in the picture. So clinician or oncologist doesn't need to rely on path. Just open and do a raster scan, and then it's possible to see whether the molecule, meaning cancer specific diagnostic marker, is visible, or what is the surgical margin. So I think time is up, so I'll end the lecture here itself. So I'll come to my acknowledgement slide. So this is my group of students and then different funding agencies which actually made it possible for me to do all this work which I generally love. And I'll end with one information to you. Actually, mass spectrometry, whatever I taught you, is still in review stage, in publication stage. There are very few books are there, especially imaging or hydrogen deuterium exchange, or native mass spectrometry. A native mass spectrometry, so you will not get any book and anything, not even good lectures also. So in the Indian government, Ministry of Education has a portal, NPTEL, you know, perhaps National Program for Technologically Enhanced Learning. So there are different professors from either IITs and other places also, I guess, meaning teaches different courses. So this year I am teaching this course. It has been already up. The class will start from January. Remember, all these courses is a government initiative. It is free. No single penny you have to pay until unless you need a certificate. So you have to write an exam, and there you have to pay to the government 1,000 rupees for that exam. Else, if you want to just don't want to credit, just edit it, meaning audit course, then you don't need to pay anything. And this is the link if you want to circulate among your students, or some of you are interested to learn the technology, then I have you can just join this. So thank you all for listening to me.
Thank you, sir.
Okay, thank you very much, sir. So now I am requesting the participants to put your question in chat box, or you can ask sir directly.
May I ask?
Yeah.
Yes,
Question.
yes, please.
So thank you for a very detailed lecture, and it was you made it very easy to follow. What I wanted to ask is, sir, I read an article recently where I learned that bone marrow stem cells have been harvested from patients suffering from sickle cell anemia. And then genetic mutation was done on it using that CRISPR-Cas technique.
Yes.
And then to make fetal hemoglobin. And then they re-administered those mutated cells into the patient again.
Yes.
So can this type of gene
Yes.
editing completely cure the disease from its
Yes,
source?
possible. It's possible. That's what is coming, and I think FDA has given clearance to gene editing issues 2-3 years back in US. But the thing is, it's highly expensive. Meaning, if you think of bone marrow transplantation, it's a matter of 50 lakhs. Now, you
I see.
think of Indian scenario, how many families would be able to pay that much? And there also risk, a huge risk, because immune compromisation has to be done. So there, even a cold cough can kill the patient. So yes, it is coming, but I hope that success rate, if it increases, then patient will definitely benefit in future.
Right, sir. But it is not cost effective at all. So that
No,
has to be
no,
taken care of
no, no.
also.
Yes, tremendous cost.
Thank you, sir. Thank you very much.
Okay, sir. I have some question in chat box, so I'm just reading one by one. So
Mhm.
first question is hydroxyurea used to increase fetal hemoglobin.
Yes.
One.
So that's the only treatment is there in sickle patient. But as you know, that hydroxyurea itself is a toxic substance. So you can marginally increase fetal hemoglobin, not too much. And that is being not only in case of sickle, but also in case of thalassemia patient. See, I was mentioning at the beginning that in case of thalassemia, say beta thalassemia, so those patients can't produce beta globin chain, can't synthesize beta globin chain. So their alpha to beta 2 doesn't happen. Now in that place, if you replace hemoglobin by alpha to gamma 2, which is fetal hemoglobin, then they can actually survive happily. They should be able to survive. May not be hard physical task they should do, but they can survive. That so this increase in fetal hemoglobin is really a challenge. And yes, with hydroxyurea it's possible, but hydroxyurea itself is a toxic, so you have a limitation. But that is only therapy. That is only therapy available effectively.
Okay, so the next question is from Nagonita Ghosh. Sir, are there any diagnostic tests available now in our country to detect any such hemoglobinopathy as you explained in fetus?
No, hemoglobinopathy diagnostic test is there, always there. Meaning, see, generally, in case of newborn baby, just by heel prick, one drop of blood they collect in blood spot, and then they characterize it through conventional method. But mass spectrometry hasn't come into the diagnostics yet, I think. Only I knew in NIMHANS, Professor Rita Christopher, who is retired now, she actually used to use mass spectrometry for different metabolites disorder, but not hemoglobinopathy so far. So mass spectrometry based detection hasn't come.
Okay, the next question from Manushi Mondal. Sir, after surgery, whenever the biopsy report shows that the portion was cancerous, doctor asks for the IHC test of the tissue block, and it is costly.
Immunohistochemistry test.
Yes, sir.
So
So,
immunohistochemistry is another method. Yeah.
and you said, sir, that it is not perfectly believable.
No, no, no,
Then
no.
what is your suggestion
I
and
didn't say
method based?
I didn't say perfectly believable. That is wrong. All I am
Okay,
saying is that is gold standard method, not IHC. Just histopath. HNE staining is a gold standard method across the globe. And then in IHC, I didn't explain it because of time constraint. Let me tell you, in IHC you label specific proteins instead of just cellular architecture, which you see in HNE staining. In IHC you see protein markers, and then there are 5-6 markers that you can see at a time uh, by fluorescent labeling. But one problem is, yes, costly, but specificity wise, yes, it is specific to protein, but because of non-specific binding, because of antigen antibody non-specific nature, false negative rate is pretty high. So that is also there, but there are markers that is available for many disease characterization. All I tried to say to you that what if we have a situation which will confidently give you the result that 0 false positive or 0 false negative. That's where mass spec might come someday. That's the message I tried to give.
Okay, now
Okay, thank you, sir.
on Mr. Chaudhary, madam, please, you can ask your question.
Thank you, sir, for your very illuminating lecture. Sir, I have one query that hemoglobin level is normal in the body, but serum iron level and the saturation of transferrin is also very low. Can you please explain how this situation can be controlled, or what the health effect it can cause?
Sorry, I don't know because I am not a clinician.
Okay, sir.
I'm not a clinician. As sir mentioned at the beginning, I'm from physical chemistry background, but have been working with clinicians, being fortunate to work with them, learned many things from them over the time. But yes, I don't have answer to this question. Sorry.
Okay, sir. Okay. Your presentation was very nice, and the way you are explaining the O2, CO2 saturation and hemoglobinopathy, I was just very pleased. Thank you, sir. Thank you.
Thank you. Thank you.
Okay, so Dr. Namrata Roy, please, you asked your question directly to sir. You put some.
Sir, I have already asked that question. I just you can ignore that message. Thank
Okay,
you.
okay.
Okay.
Fine. Okay, so any. Okay, if not, then I'm just formally putting our vote of thanks. So, sir, it was really a wonderful lecture that you explained, and I can resist myself to say that I am also from. I've done my PhD from the Biological Science Department in Azad, Kolkata, under Dr.
Okay.
Rupak Datta.
Oh, achha, you are Rupak student.
Yes, sir.
Okay.
So,
He's my
thank you,
good
sir.
colleague.
So, yeah, exactly, sir. So now, on behalf of our institutes and the University of Kolkata and all the participants in this virtual platform, I extend our heartfelt thanks to Dr. Amit Kumar Mondol for his outstanding and highly engaging session. Sir, we are really grateful to you for sharing your time and your expertise with us. Thank you very much, sir. So now we are moving to our next session. So I think.
I'll log off. So can
Yes,
I?
sir. Yes, sir. You can now log off. Thank you very much, sir.